Okay, so we'll get started. So firstly, thank you all so much for coming. So my name is Dr Helen Coulshed, and I am a senior lecturer in chemistry education at the Department of Chemistry at King's, and I'm one of the people that you'll spend an awful lot of time with if you come and study chemistry here. That's a good thing, by the way. So I am here, I've got the very massive privilege of introducing you to kind of why we're here, so talking a little bit about the Daniell Lecture, and then I'm going to introduce both of our speakers. So our first speaker is Desmond Koomson, who is from the chemistry department. So he's one of our postgraduate students. So he's studying for his PhD at the moment, and he'll be giving a talk about the work that he's doing and will continue to do during his PhD. It's super exciting. I'm confident you'll enjoy it. And then I'm going to talk a bit about our Daniell speaker, who is Professor Sir David Klenerman, and then I'll pass over to him to talk about the excellent work that he's done so far. The aim is for us to finish by 6, so hopefully that's what you had in mind. So yeah, we'll get started. So, why Daniell? So probably a lot of you haven't necessarily heard about him. You wouldn't necessarily learn about him in chemistry at school. But he was someone that was born in 1870, but like most of you, he was very interested in the world around him and understanding like how it works and how we can measure it. So a lot about science is being able to measure something, having a measurable change, a measurable impact. And so we actually invented lots of ways to do that. So some of these are listed here. So the hydrometer, so measuring atmospheric humidity, but also, who doesn't like a bit of pyrotechnics? So he's also looked at pyrometers, so being able to measure extremely high temperatures, particularly in furnaces. And I'm sure some of you might have made creme brulee before. So he made one of the first kind of safe blowtorches, or welding if that's a secret hobby of yours. So King's College London was founded in 1931, and he was the first head of chemistry in our department. But he was a passionate teacher, so he was very interested, as you might have been able to tell, in atmospheric chemistry, but he was so interested and dedicated to teaching, actually he ended up giving up that research area and focusing predominantly on teaching. But the good thing about studying in a department is you often meet other people when you're studying and you get inspired by what they do. So one of his peers was someone called Michael Faraday, and with him, as well as the fact that electricity and electrochemistry was becoming more and more kind of prevalent and interesting, he sort of switched focus from atmospheric chemistry over to sort of looking at electrochemistry, and realized quite quickly that he needed a kind of reliable, constant battery. So that's what he invented. So that's the Daniell cell. So that was the first kind of constant current battery, and that's the kind of, the first part of what you now have in all of your laptops, phones. Because of that chemistry, that idea, that's why any battery is possible, okay? Obviously, like anything, he was a human with other people that he loved in his life. So he had 7 children, very productive, 5 daughters and 2 sons, but his wife passed away around the time of his discovery, and that coincided with him losing all of his notes. So it wasn't like a laptop we can save it, so he actually lost his notebook with all of his research in. Generally quite devastating, but he then repeated all of the research and was able to kind of share it with the scientific community in 1834. A little bit of a weird story to end on. So he was at a Royal Society meeting a day after his 55th birthday and died suddenly of a heart attack. But due to the breadth and impact of the research that he'd done, the science that he'd looked into, they awarded him every possible medal from the Royal Society upon his death, which is pretty extreme, but also a good recognition for that. So therein lies why we're here, well, one of the reasons why we're here today, to kind of talk about chemistry and celebrate his life and research. So I'm going to hand over to the wonderful Desmond now, who's going to talk about his research. I think this is working. Yes, it's going to work for you, not for me.
Hello. So good afternoon. I'm Desmond, as Dr. Helen Coulshed introduced. I'm a PhD student in chemistry department, specifically doing electrochemistry. And before I begin, I want to thank the organizers for allowing me to talk in this Daniell lecture. And thank you for coming as well. I hope you had a good time here. And I hope this talk will be very interactive as well. So I'll be talking about chemistry, my research journey, why I decided to do chemistry. And we know from subatomic particles to galaxies, chemistry is all around us. And investigating how nature behaves from molecular or atomic level has really advanced our lifestyle, even in technology and health as well. So that was something interesting to me when I was at your age, high school. So I decided to do biochemistry in Ghana, in University of Cape Coast, to investigate about the food that we eat, how do we get energy from the food that we eat. And so during my first year biochemistry, I went to this lecture, and they spoke about glycolysis. So we all know about food that we eat. It contains carbohydrate and stuff in it. And how do we get energy from the food that we eat? And through that lecture, I was able to understand how a simple molecule like this called glucose is able to pass through this pathway called glycolysis and Krebs cycle to give us the energy that we are able to have as humans. And that was really fascinating to me, because it gives me a better understanding about the world that I live in and how energy is being produced from the normal food that I eat. So during my final year, I decided to do more research into food. I don't like food that much, though. Yeah, but in Ghana, we have this shrub called turkey berry. How many of you know about turkey berry? I think it's not common here in the UK. Yeah, but it's really common in Ghana. We use it for everything. So my mom used this turkey berry for cooking, whether it's stew, soup, any food, turkey berry. If you are sick, turkey berry. So we do everything with turkey berry, and I really hated turkey berry. Yeah, I really didn't like it because, and I asked my mom, why do you always use turkey berry for food? And it's like, oh, it's healthy to give you, like to boost your health. When you're sick, there are some health benefits in it. I said, you're not a scientist, so how do you know that this has health benefit in it? And so my grandmother told me, and her grandmother told her. So I was like, that cannot be true. And I wanted to prove my mother wrong. So, so I was really happy when my supervisor at that time told me, hey, Desmond, I want you to do an investigation to see the health benefit of turkey berry. I said, this is it. Is this the time I can prove my mom wrong about this turkey berry? So I investigated the phytochemicals. And phytochemicals are just chemicals that we get from plants, and they are bioactive, and they have some health benefits from it. So I looked at the phytochemicals, this 5 main phytochemicals, and also vitamin C. I hope you all know about vitamin C and how important it is. So first of all, I wanted to see if these chemicals are in turkey berry, and unfortunately for me, they were. So, and these phytochemicals have a lot of health benefits, and some of them are antioxidants, and they can also combat diseases, even including cancer as well. And some researchers have been able to use these phytochemicals to study cancers in rats and mice as well. So all this while my mom was wrong. It was right, and I was wrong. And that really intrigued me, that if I can be able to investigate and find these things in the normal food that we eat, can we also apply it to the systems, the things, the energy that we find in food? Can we translate it into the normal energies that we use around us? And before I started my master's, I was really intrigued about the Nobel Prize in Chemistry in 2019, won by this amazing 3 Nobel laureates. That's the development of lithium-ion batteries. How many of you know about lithium-ion batteries? Oh, don't be shy. You just raise up your hand if you know about lithium ions. So how many of you have phones or laptops? So your phones, your laptops, most of them contain lithium-ion batteries. And it's not working. Okay, let me use this. So there's just a schematic diagram of lithium-ion battery. And so basically, the lithium ion, which is positive, just moves into the membrane here to the cathode over here, and the electron moves through the anode, through a circuit. So the movement of this electron creates current, and through that we're able to power bulbs and even our phones and the cars that we have. So there's a car battery. There's also a lithium battery for laptop. And this is how fascinating chemistry can be. This is an element in the periodic table that you've learned at school, right? And through this simple thing, we're able to have an amazing application. And that was really fascinating to me. So I asked this question: how am I able to use the same concept in a sustainable way to generate electricity, and not generate electricity, but also to break down food or waste material in something similar like this? So I did my master's in China in 2018. So during my master's, I created this, I designed this cell called a microbial desalination cell. And all that this cell does, it's the anode chamber here contains wastewater. And the middle chamber normally contains seawater, but this time around I use ammonium chloride, which is also a salt. And you can be able to retrieve ammonium from this. And here contains a reducing agent that's able to accept electrons. So the three main things that this cell does is it breaks down, it treats wastewater, it desalinates or cleans salty water, and it produces energy, only by using microbes. So you have microbes in here that breaks the carbohydrates or the glucose that we have here. So again, using normal food waste, you can be able to add microbes to this. They give electron to this, and it produces energy. And as I want to make it more interesting, I added another cell to it. So I can give this energy, this electrical energy that's being produced here to the cell, and be able to reduce heavy metals from here. So now 4 things are happening. I'm treating wastewater, producing ammonium, or cleaning seawater, and also producing energy to be able to remove heavy metals from wastewater in the system as well. And that's how, from the simple understanding of carbohydrate and how it works and how our system is able to break down this carbohydrate or these simple sugars like glucose, we'll be able to apply it in some systems like this. So moving from my master's, I came here to, now instead of using microbes, how does microbes break down these simple sugars? And how are they able to produce this kind of electrons that we see here? So I decided to do, this thing's not working. Okay, so this is just a video of the whole system over here. So this is a microbial desalination cell. And this over there monitors the power generation. So this system over here is where the heavy metals are being reduced. And if you want to read more about it, we're able to publish 2 papers from that, so you'll be able to read more about that as well. And so to go further with that, I decided to look at enzymes, because our body contains enzymes, and actually these enzymes are those that do the work that we see over there. And how many of you know about activation energy? Oh, I like this class. Good. So we all know about this, what is happening here, right? So these reactants need a minimum activation. They need to the minimum activation energy needed for these reactants to react to produce these products here. Without the enzyme, they have to have this activation energy here, but with the enzyme, it reduces this activation energy, right? So a typical example is a glutathione reductase. This enzyme it uses this compound called NADP plus and NADPH to reduce this molecule over here. And this molecule that's called glutathione is an antioxidant. What does this molecule do in our system? All that this molecule do is it reduces these radicals in our system. So it acts as an antioxidant. So it helps our body to remove radicals. And radicals are molecules or atoms with unpaired electrons, and they are sometimes very harmful to our system. So if you have this enzyme and you're able to reduce this glutathione to this reduced glutathione, it helps your system. But are we able to translate this again into a normal system where we can be able to replace this, because this is very expensive to produce in the industry? Can we replace this and use this same enzyme in a cell, as I've showed you earlier, where we'll be able to reduce this and also produce energy just using our body temperature or heat waste? So that's what my current research is about. I'm trying to replace the NADPH, which is really expensive to produce, with a simple compound called Vialogen, which is normally a herbicide. So it's very easy to produce or synthesize in the lab. And it also takes an electron from the electrode over here, gives it to this enzyme again, and it repeats the same thing that the NADPH is supposed to do. So getting the understanding of how chemistry works, we'll be able to replace something that occurs naturally in our system with something that can also be sustainable as well. And hopefully the end result, the end result will be a thermocell like this, where whilst these Vialogens are reducing these enzymes to produce these compounds or any other compounds, we get electrons being produced based on temperature difference. So the application will be from pacemakers. How many of us know about pacemakers that we use? So you can have a system where your body, the difference in your body temperature can produce electrons to help power your own pacemaker that you are using. Or can be used in very huge industries where, like pharmaceutical industries, where they produce compounds using enzymes, and we can tap their own energy, their own heat energy that they are producing. So this system can be a self-sustaining system. And this all became possible because of how we understand chemistry. And I followed this journey because I was interested in something called glucose, a simple molecule glucose. And through that, I hope that you'll also be interested in chemistry as well. Thank you.
Does anyone have any questions for Desmond about his science or anything that he just spoke about? Yes, okay, I'll be coming right up.
Thank you. That was really, really fascinating. I didn't understand a lot of it, but it was really interesting. I was wondering, how much choice do you have over your PhD project? Like, how much say do you have over what you get to research and study?
Okay, that's a really good question. So here at King's College, as a PhD, even though you have two supervisors, you are free to use the instruments. You are free to make purchases that you want to make based on the funding that you have. Yes, they need to be approved, but so far as whatever you are doing is in line with the research you are doing. For example, coming to King's College, the project was already there that I decided to choose which project that I want to do, and based on that I was interviewed on, and I decided to work on that because that project that I'm working currently is really interesting to me. So I decided to do that, and that's what I'm doing. So I have the freedom to use any instrument or make any purchases that I want as a PhD student. Yeah.
What is your mum think of your research on kind of prove her right?
I hope she's not watching this. Yeah, actually, she was really excited about it. Yeah. I told her when I did that research on the turkey berry, and my mom is always trying to, like, yeah, I told you so. Yeah. But my mom and my dad has been a very great, encouraging people in my life. They were able to cheer me towards the research I'm doing currently because they knew how important in science and specifically chemistry is. So anytime I speak, so normally I share my ideas with them when I'm doing, and they just push me on. Yeah, I sometimes disagree with them concerning certain things, but always, I don't know how, but parents are always right. Yeah.
Thank you. Just a simple question. What is the biggest challenge from your research?
My research journey, or just
I think the last one, the what is it, electrochemical something? Yeah.
My current research.
Yeah, your current research, yeah.
That's a really difficult question. I would say the biggest challenge will be trying to understand how these enzymes work, especially because all this while I've been working with microbes, bacteria as well. So trying to enzymes, and that's really amazing when we are working with them. They are proteins, and they can denature in certain mediums. So trying to understand how they work and how my mediators, the biologens I produce, is able to interact with them. That's one of the biggest challenges that I have, yeah.
Okay, we have time probably for one more question. Okay, Desmond's going to be in the room for the next talk as well, so if you want to grab him afterwards and ask a question, please do that. But yeah, thank you very much, Desmond. It was amazing.
Thank you.
Okay, so we're now going to move on to our invited speaker, so Professor David Klenerman. I'll talk to you a little bit about his background before passing over to him to speak. So a little bit more about David's background. So as you can see, he did his undergraduate degree at Christ's College in Cambridge a little while ago, and then got his PhD also from Cambridge in Churchill College under the supervision of Professor Ian Smith. He then went across the water to the US to study at Stanford with a Fulbright scholarship, or as a Fulbright scholar, before returning to work in the UK, and he worked for BP, so British Petroleum, for 7 years. He then returned to Christ's College, back to Cambridge in '94, where he met his colleague, Dr. Shankar Balasubramanian, and they worked very closely together on the next generation DNA sequencing, which will be, I think, the main theme of this talk. So yeah, let me hand over to you now. Thank you very much.
Great.
More lively than this one.
Okay, great, brilliant, thank you.
You're welcome.
Great, thank you very much for that introduction. So I'm sort of at the other end of the spectrum from Desmond, since I'm sort of at the latter stages of my academic career. But I think what I wanted to do is also make the case of why I study chemistry and how transformative chemistry potentially can be. And that's largely by telling you about the story of how we came up with this way of doing next generation DNA sequencing. So I'm going to really spend a lot of time explaining the bits that led to that idea, and then why it's been quite transformative. So that's really the idea behind my talk. So I'm going to say a little bit more about my background. I'm a physical chemist, so I'll show you a little bit about what physical chemists do. I'm going to introduce to you single molecule fluorescence, this very exciting idea that you can actually watch individual molecules in live cells. And then I'm also going to introduce you to super resolution microscopy, which is a way of getting very high resolution images of structures. Then I'm going to show you how we're developing and applying these methods to study protein aggregation and neurodegeneration. And I think you'll see that we like building new instruments, but I'll show you a lot of the instruments that we built during this time. And then I'll introduce to you DNA sequencing and next generation DNA sequencing. And the main theme is how new physical methods can enable new science. So this is my supervisor, this is Ian Smith. And this is the apparatus I built for my PhD. So I made all this glassware. I learned how to do glass blowing. So this allowed me to do gas handling. And then the gases were fed into this reaction chamber here with this big pump. So I worked at very low pressure. And the reactions that we studied produced vibrationally excited molecules. And we were interested in looking at the infrared radiation that they emitted. So this is a little bit more detail. So the gas came in, there was a microwave to convert hydrogen into hydrogen atoms. I made ozone and reacted hydrogen atoms with ozone, and that produced OH and oxygen, and the OH was vibrationally excited and emitted light, and we were interested in how much infrared radiation that produced. Like's been said, I then went to Stanford for 2 years, where we tried to use lasers to cut chemical bonds very specifically, but by exciting them with a large amount of visible light. Unsuccessfully, we were quite good at heating them up, but we couldn't cleave the bonds specifically. And then I came back and worked for BP. So this is a picture of me, this is a younger version of me in the lab. And this is the laser lab. So there's a green laser that pumps a dye laser here. So maybe you can see the green lights here. The dye laser emits yellow light, and it's tunable. And this light was very short pulses, so the picosecond versions, picosecond duration of light. And this was amplified by this laser here. Then it went down the laser table to the sample that sat somewhere down here. And the idea of these experiments were really to see what was going on at the surface of metals, either when the metal was corroding, to understand how the corrosion inhibitors that BP used worked. So they used vast amounts of these on the Alaskan pipeline, for example, to understand how lubricants that they use in the cars work and prevent damage to the parts that move. And then in 1994, I went back to Cambridge. And basically, once I arrived in Cambridge, the most exciting thing that was going on in my field was the idea that you could detect single molecules. And so most of my research has been exploiting and developing new ways of imaging single molecules and applying it to biology. So the main idea behind single molecule fluorescence is you have a dye molecule, and that dye molecule will attach to the molecule of interest, and we can excite the dye molecule with a green laser, and then it will fluoresce yellow. So we can nicely separate the green excitation laser from the yellow light that the dye emits. And we can have one dye molecule, and we can excite it again and again and again, and it will emit light, it will emit a photon. And we can do this roughly 1 million to 10 million times before the molecule blows up in its excited state and no longer emits a photon. But that's enough. We get this amplification of a factor of 1 million to 10 million, and that's enough to enable us to study single molecules, as I'll show you in the next few slides. So when we do single molecule experiments, we typically work at very low concentrations, typically done at picomolar concentrations. And under those conditions, there's typically one or less molecules in the volume that we're probing at any one time. And also, very importantly, if we do experiments with single molecules, it's really the ultimate in miniaturization. And this is one of the ideas that we exploited to develop next generation DNA sequencing. So I just wanted to just make sure we're all on the same wavelengths. But so I use lots of words like nanometers and things just so you understand what those are. So this is an ant. This is a human hair. This is a few hundred microns in diameter. This is a cell. This is typically 20 microns in diameter. This is a bacteria, less than 10 microns, a mitochondria, the powerhouse of a cell, a micron. And all these things we can visualize very nicely using light. But there's a fundamental limit, the diffraction limit, below which we can actually observe things, but we can't make out their structure. And that's around 200 nanometers. But there are lots of things we'll be interested in visualizing with light that are smaller than 200 nanometers, such as viruses, proteins, or I'll tell you about protein aggregates. And so one big advance in my area was a method that was developed to be able to break this diffraction limit and observe objects using fluorescence that are smaller than 200 nanometers. So I haven't found a very elegant way of doing this, I'm afraid. So this is a laser lab. Since I can't take you into a lab, I wanted to show you what a lab looks like. So we're in the dark, and we build our instruments on these very large laser tables. It's all under computer control, so everything's very automated. Maybe I'll just stop while these people come in. So Helen, shall I restart or?
If you can switch to the microphone, because I think the people online are struggling to hear you, if that's okay. Sorry.
Okay. Do you want me
Yes, probably. See, that's quite a large group of viewers. Sorry about that.
That's okay.
Yeah, if you're happy to, yeah.
Okay. It's just the room with double the audio.
Yeah. Okay, if you can get seated as quickly as possible. David's kindly said that he'll restart the talk. So do your best to find somewhere to sit. Thank you. There's plenty of space at the front. Okay, I think we're all emotionally and physically ready to get going. Is that okay? Yeah, magic. Okay, I'm going to hand back over to David. Thank you very much for starting again.
So I was going to tell you about why I study chemistry and particularly share with you how, with one of my colleagues, Shankar Subramanian, we came up with a new way of sequencing DNA very rapidly. So the way I've structured this talk, I'm going to tell you a little bit about my background. I'm going to introduce you to single molecule fluorescence and super resolution microscopy. Then I'm going to show you how we developed and applied these methods to study protein aggregation and neurodegeneration. So these are diseases such as Alzheimer's disease and Parkinson's. And hopefully you'll see that I'm a physical chemist, that physical chemists like building new instruments that can measure things that no one else can measure. And then I'm going to spend most of the talk explaining how the various items I've shown you, single molecule fluorescence, can be used to develop a new way of sequencing DNA, and how surprisingly impactful that was, and how it's being used today. And the main theme of this talk is really how new physical methods enable new science. So this is a picture of the apparatus that I built when I was a PhD student. I worked with Ian Smith at Cambridge. And so you can see all the glass blowing that I did for gas handling to handle the gases. And these gases came to this reaction chamber here, and we produced radicals, and that produced molecules that were vibrationally excited. And this device here was a device to look at the infrared radiation emitted by these molecules. And this is a little bit more of a close-up of the reaction chamber. So the gases came in, we converted hydrogen with a microwave into hydrogen atoms, and then that was reacted with ozone that I also made, and that produced OH in a vibrationally excited state. And I had these mirrors here that collected that infrared radiation and sent it to my infrared spectrometer that I also built, and measured how much infrared radiation was being produced by the reaction. I then went to the States for a couple of years where I tried to cleave organic molecules at a specific bond and do sort of what was called laser selective chemistry. That was unsuccessful. We were very good at heating the molecules up with the laser, but we couldn't cleave the bonds and cut the molecule where we wanted. And then I came back to the UK and worked for BP, British Petroleum. So this is a younger version of me in the lab. This is my laser lab. So we'll have this green laser that pumped this dye laser. You can see it's emitting yellow. There's an element in here to tune the wavelength. This laser produced very short pulses of light that were amplified by this pulse laser here. And then the light was sent by a whole bunch of mirrors down to the sample that was down here. And the idea of these experiments were to look at what was going on the surface of metals. BP were particularly interested in how the corrosion inhibitors that they used on the Alaskan pipeline actually work, so we could develop better corrosion inhibitors, or how the lubricants that they sold in their oils actually work to devise better lubricants. And then in 1994, after 7 years at BP, I came back to Cambridge. And one of the big advances in my field at the time was the ability to see individual molecules. So chemists are really interested in molecules, but we typically study billions at the same time. But methods were developed to actually look at individual molecules diffusing around or actually undergoing biochemistry in living cells. And so the principle of this is we have a dye that's attached to the molecule of interest, and we excite it with a green laser, and the dye molecule emits yellow light. And I can do this cycle again and again and again until the molecule undergoes an irreversible reaction in its excited state. It essentially blows up and no longer emits fluorescence. But a good dye molecule can undergo this cycle 1 million or 10 million times, and that's essentially the basis of single molecule fluorescence. One molecule can emit 1 million to 10 million photons, which are easily detectable using sensitive cameras. You can actually even see single molecules in a dark room with your eyes, and that enables us to study single molecules. So we typically work at very low concentrations, typically picomolar concentrations, where there's less than one molecule in the probe volume at any one time. And it's really the ultimate in miniaturization, and that's important when it comes to developing the next generation DNA sequencing. So just to calibrate us all, this is just to make sure we understand the length scales. I'll use words like nanometers. I just want to make sure you know what these are. So this is an ant. It's a few millimeters. A human hair, a few hundred microns. A mammalian cell, 20 microns. A bacteria, a few microns. Mitochondria, the powerhouse in a cell, submicron. All of these things we can detect with light, and we can resolve their structure. But there's a fundamental limit, the diffraction limit of 0.2 microns, 200 nanometers, below which we can't make out the structure of objects, even though we'll be very interested in them, such as viruses, proteins. I'm particularly interested in protein aggregates or smaller molecules. So one of the other big advances in my area was the ability to break this diffraction limit and do super resolution imaging. So this is where I was before we doubled our audience. So I just wanted to show you what a laser lab looks like. So we work in the dark. We have these big laser tables. Everything's under computer control. So it's all very automated. Sorry, this video is slightly slow, so if it gets very boring, I'll speed it up. And then we have an array of optics that was less interesting on our power supplies, which is probably not such a good thing. And here we have an array of optics. We have devices in here to control the power of the lasers. These are the diode lasers. We have filter wheels to either let the light through or not let the light through. And then this light is all combined and goes over to a microscope, which is coming up in a minute. And the light goes up through the objective of the microscope, excites the molecules. They undergo multiple cycles, like I described, of fluorescence. And then we detect that using a sensitive camera. So we're just about to move around to the microscope, hopefully. Right, so here's our microscope coming up. So it's an inverted microscope, and this is a sensitive camera that we use to detect the fluorescence. So this is what the lab looks like. It's very colourful. Purple laser, Cambridge blue laser, green laser, red laser. We combine all the light with mirrors, and we shoot it over to the microscope. At the microscope, the light undergoes a total internal reflection at a glass cover slip. And so hopefully you might have learnt about this at physics. If you get the angle right, instead of transmitting through the glass, the light undergoes total internal reflection. But there's a evanescent field which penetrates typically about 100 nanometers above the cover slip. And this allows us to excite molecules that are present in the cell membrane. So this example is a T cell, a white blood cell. We've got T cell receptor, which is the receptor responsible for identifying cancer cells. And we've labeled that with an antibody that has a dye attached to it. And then you can see these molecules diffusing around on the cell surface in real time. So we're interested in how the molecules are organized on the cell surface and how they reorganize when the T cell recognizes a cancer cell and mounts an immune response. So this is certainly was when we first did this, very exciting. We can actually see single molecules on a live cell. This is another example. So these are molecules of DNA, and we wanted to show that each of these molecules of DNA, which is labeled with a fluorophore, is largely a single molecule. So we excite them all, like I've just shown you, and like I've explained, the molecule will cycle and produce photons, and then it will undergo this destruction and stop emitting. So if most of these molecules are single molecules, they'll emit for a period of time and then turn off. If occasionally we have two molecules that happen to be close enough together that we can't resolve them, so less than 200 nanometers apart, we'll get two photobleaching steps. The first molecule photobleaches, then the second molecule. So this is the example for this one with the double dots. And very, very occasionally we have three molecules in the same 200 nanometer region, and we get three steps in the photobleaching curve. So by doing these type of experiments, we could show that about 95% of these molecules were single molecules, and we're really studying single molecules. So the other thing I wanted to show you is how we break the diffraction limit. So this is an image of something, and you can make out the structure, but it's blurred by diffraction. So the idea is we're now going to organize that we just look at single molecules. We have a single molecule here. It's also blurred by diffraction, so it shows up as a spot that's 200 nanometers in diameter. But I now know this is a single molecule, so it must be at the center of this spot. So I can replace this blurry spot with this spot where I've localized a molecule with much higher precision, typically 10 nanometers or so. And then what I have to do is repeat this process again and again and again, and I can build up a super resolution image of the object of interest, now with a resolution that's typically about 20 nanometers. So this was developed in early 2000s, and in 2014, Eric Betzig and Morna and Stefan Hell won the Nobel Prize for chemistry for developing this methodology. So one of the problems we're interested in is what causes Alzheimer's disease. So this is a picture of the brain of someone unfortunate enough to have Alzheimer's disease. And what you find is these deposits of a protein called amyloid beta outside the neurons. And you see these tangles of tau inside the neurons. But it's what's known in the field, it's not these macroscopic, insoluble structures that are causing all the damage in Alzheimer's disease, but it's the much smaller soluble aggregates, the smaller aggregates that grow to become these plaques and tangles that cause all the damage. And these are very difficult to study using conventional methods, which is why we decided to use our single molecule techniques to look at these aggregates. And there's a third protein I'm going to discuss called alpha-synuclein that forms inclusions, aggregates in Parkinson's disease. So we're interested in developing the methods to determine which aggregates are toxic and their mechanism of toxicity so we can rationally design therapy for Alzheimer's disease. And we really wanted to study the real species that cause the disease in humans. So this is an experiment that one of my PhD students did that was very exciting. I didn't actually think this would work when he tried it. So what we did is there's a dye called thioflavin T, which is non-planar in solution and non-fluorescent. But when you have one of these aggregates, they have a beta sheet structure, which means that the dye can become planar and bind inside the beta sheet structure, and it becomes highly fluorescent. So what we did was take some cerebral spinal fluid. So this is the fluid that bathes everyone's brain, and you can get at it by taking a lumbar puncture. And what we did was take some of this cerebral spinal fluid from a patient unfortunate enough to have Parkinson's disease, diluted it tenfold, added this dye, and put it on one of our microscopes. And much to my amazement, because I thought we'll just be swamped by background signal in the cerebral spinal fluid, we saw these small spots. Each of these spots we think represents a protein aggregate. It's probably smaller than 200 nanometers. And we realized we could look at the individual aggregates present in a human sample. And it raised an awful lot of questions. What are these aggregates? Are they alpha-synuclein? How do the aggregates change as someone develops the disease? And so a lot of our work since then has been exploiting this discovery, trying to work out what are the real aggregates and how they change in neurodegenerative disease. So this is just one example. This is cerebral spinal fluid from a Parkinson's disease patient. And this is a 100 nanometer scale bar. And this is the image of the aggregates of alpha-synuclein inside the cerebral spinal fluid. So you can see that these small round aggregates. What we found is they're bigger aggregates and more of them in the serum of a patient with Parkinson's disease. And then we found that there are more of these big aggregates in someone with Parkinson's disease than someone who doesn't have Parkinson's disease. And so we're quite hopeful this might be an early diagnostic for Parkinson's disease. And we're similarly doing things for Alzheimer's disease. And we'd hope that maybe we'll have a diagnostic for Alzheimer's disease that can be used in the blood. And also, which has been on the news quite a lot, the football players or rugby players who get repetitive bangs on their head and get early onset dementia, we might be able to see the changes in the aggregates in their blood to be able to work out before they get the disease that they've actually undergone some sort of event that predisposes them to get Alzheimer's disease. And one would hope maybe some of the treatments might be more effective if given to them at that stage. So these aggregates, we think, cause inflammation. So we built another instrument to look at how they cause inflammation. So what we did with this instrument is we have a light that is focused to a thin sheet, and it comes in and interrogates the top of the cell. And I have a fine nanopipette, so this is typically has an aperture of about 100 nanometers, and we can bring it very close to the cell surface, and we can shoot aggregates of beta amyloid, in this case, out onto the cell surface, and then we can watch the signaling of the inflammatory response inside the cell. So just to show you, we're very good at shooting molecules and putting them in defined places. This is a real-time video of us delivering a glycoprotein onto a glass surface. So we can make pretty patterns. And so what we did is study this process where a toll-like receptor on the cell surface responds to these aggregates we deliver from the nanopipette. And what happens is the toll-like receptor 4 dimerizes and then forms this midosome, which is made up of 6 MyD88 and these other proteins. And so what we did in this cell is tag the MyD88 with GFP, and what we see before we turn on, before we trigger is that the MyD88 is all over the cell. But what's going to happen when it forms a midosome is we'll see these spots form on the cell surface. And once this midosome's formed, there's this biochemical cascade, and then this protein, NF-kappa B, enters the nucleus, and it gives rise to the production of pro-inflammatory cytokines. So this is a very early event that we think is important in Alzheimer's disease. Oops. Let me just go back. We missed the video. So hopefully you can see these spots form in real time as we form these midosomes on the cell surface. So the rest of the talk, I'm now going to focus on DNA and how we sequence DNA. So almost all the cells in our body contain DNA, and the information stored as a code, we've got 4 bases: adenine, which is A, guanine G, cytosine C, and thymine T. And the order of these bases really determines all the information for building and maintaining an organism. And it's a code, so the DNA can be copied from new cells because a single strand, since we know that A always pairs with T and G always pairs with C. So this is just a little bit more detail. This is C paired with G, it forms 3 hydrogen bonds, and T pairs with A, it forms 2 hydrogen bonds. So DNA is fundamental to life on Earth, and if you want to work out the sequence of all these bases, you need a sequencing technology. And why do you want to sequence DNA? Well, like I said, you want to understand life, and you want to understand the differences between humans. We differ between 1 base in 1,000. And then if we could do this, we could find the cause of diseases. We could detect if people were going to develop a disease. We could diagnose what disease people had. We might be able to prevent people getting the disease before they had any symptoms by sequencing them and intervening before they got the disease. We could develop better treatments, and we could monitor how effective the treatments are. But the problem is that all of us have 3 billion bases in our DNA. So you need a sequencing technology that can decode these 3 billion bases. So this is how it was done before we came up with our method. Maybe you'd be familiar with this. You have your template of DNA, and we're going to remove 1 strand. So we have a single strand of DNA, and we have a primer. And we're going to add the nucleotide triphosphates to copy the DNA. But we're also going to add these terminated bases. And these bases are color-coded. A is red, T is green, G is orange, and C is blue. And we're going to add an enzyme called a polymerase that's going to make millions of copies of the DNA. And occasionally, the polymerase will incorporate one of these terminators, and there'll be no further growth of this strand of DNA, and it will be tagged with a T. And so you end up with a short length of DNA with a T terminator, a longer length with A, et cetera, and then you can run these on a gel, and the short strands come out first, the longer strands come out later, and from this you can work out the sequence of bases in the DNA. So in this method, it's 1 tube, 1 gel or lane or capillary, and you could typically do 500 bases per run. And this is sort of a macroscopic, the gel capillary is typically a millimeter or so in diameter. So it costs $3 billion to sequence a human genome, and that was just sort of the average human genome. It wasn't the human genome of an individual. At the time that we started this, the cost of a human genome was 30 to 50 million, and the challenge was to reduce this cost down to $1,000 or less. And if we can reduce the cost significantly, we can start to sequence individuals and see the differences between each of us, the differences that predispose us to get cancer or protect us from getting cancer. So at the time, we weren't actually trying to do any DNA sequencing. I had two postdocs, Colin Barnes and Mark Osborne, and we were trying to do an experiment where we have DNA, we have a primer to start the polymerase off, and we were trying to watch the polymerase incorporating individual nucleotides into the DNA just to see how it worked and watch it spiral up the DNA, copying the DNA. So this was back in 1995. And it was a difficult experiment because the way we set it up was we had lots of template primers all over the surface, but we could only look at one of them, and we kept on missing the polymerase incorporating the bases. So this is my colleague Shankar. This is, again, a younger version of me. This is the pub that's very close to the chemistry department. And we went here fairly regularly after doing experiments to try and work out why these experiments weren't working. And while we were having these discussions, we came up with this idea of how we could use our technology for DNA sequencing. So this is the idea behind our sequencing. We have a template primer, so our DNA we want to copy, and we started off with this short strand of DNA. And then we're going to have our color-coded bases again, and we're going to organize that there's a blocker, so we only put in a single nucleotide. In this case, as you know, we've got a T here, so we're going to have an A incorporated. And then we're going to read the color of the base, and then we're going to remove the fluorescence, and then we're going to undo the terminator, and then we're going to repeat the cycle again. So unlike the Sanger sequencing, we've got reversible terminators, so we can go around this cycle again and again and again and sequence this strand of DNA by noting the color of the fluorophores that have been incorporated. And then the other big idea was we're going to extract the DNA, put it on a surface, and have lots of molecules all about a micron apart so we can resolve them, and we're going to sequence all these molecules at the same time in a massively parallel fashion. So we've now miniaturized the whole process. These molecules are a micron apart, and we're going to image, sequence billions of them all at the same time. So the reason the method is so successful is it's very accurate because we're copying the DNA using the polymerase. It's very fast because we're doing it in this massively parallel fashion, and it's very low cost because we're using very small amounts of molecules. Originally, we were working at the single molecule level, but as I'll show you, we amplify these up into little colonies of molecules. So once we do these very short reads of the DNA, so we're going to sequence 30 bases of DNA, we can assemble all these short reads of DNA onto the average consensus sequence of DNA, and hence assemble the overall sequence of all the DNA in our sample. So all these little bits can be organized, and we can then assemble the full length of DNA. We have some problems with repeat sequences of DNA, but we can do this for most of the DNA in the human genome. So this is a calculation that we did back in 1997. So there are 3 billion bases in the human genome, and we assume that we'll image an array of 300 by 300 single molecules, and a sequencing imaging cycle took 10 seconds. So we have 10 to the 5 roughly bases, divide by 10 seconds, so we can sequence about 10 to the 4 bases per second. We can go at 10 kilobases per second. So one machine's going to take 3 billion divided by 10 to the 4 seconds to sequence a human genome, which is about 5 days. So we thought this was a great way of potentially sequencing a whole human genome in a short length of time. So what we did is found this company called Solexa. We realized we needed quite a lot of money in order to go all the way and really develop this technology. So we founded a company, and indeed we could get increasing amounts of money to develop the technology. the name doesn't mean anything. It's sort of, we were playing with ideas, single molecule, and then Shankar thought that any decent company like Xerox had to have an X in it, so that's why the X is there. This was we originally had some ideas of a DNA hairpin, so this is sort of the structure that's here. So we founded this company back in 1997, and that's what developed the ideas that I've just told you about. This is the nucleotides that Shankar designed. So we have a reversible terminator here, and we have a dye that we can cleave off. It's worth pointing out we don't end up with exactly the nucleotides that are natural. We sort of have this short chemical stump left, and we had to mutate the enzyme so it could deal with this chemical stump that's left after the cleavage reaction. And like I said, instead of studying single molecules, it turned out to be much better to amplify them all into little colonies of 100 molecules. And this massively simplified the apparatus. So this is what it looked like when we got it to work. So this is a 100 micron scale bar. So just to remind you, human hair is a couple of 100 microns. So you can see we have vast numbers of sequencing lanes. And also, hopefully you can see in this zoom, we have lots of different color spots, which is telling us about the base that's been incorporated in that sequencing round. So this is the instrument in 2006 that we built. So this is our logo, this is our instrument. This is 2021. So this instrument could do 1 billion bases per run, so that's a gigabase. The current instruments, and this is not up to date, can do 6 trillion bases. So billions, 1,000 million, trillions, million bases per run. So in 1997, the number of bases per instrument was 10 to the 5. In 2021, it's 10 to the 13, so we've improved that by a factor of about 10 to the 8. The number of sequencing lanes gone from 100 to 10 to the 10, another improvement of 10 to 8. And the cost, which is probably the most important thing, has gone down from $1 billion for a human genome down to below 1,000. And the company's now just launched an instrument that can sequence a human genome for about $200. So that's a millionfold reduction in cost. So we went from this situation, this is the cover of Science, so it's a drinks party, and there's this lizard talking to this fly, and it's saying, well, have you had your genome sequenced yet? And this is in 2009, we published the first sequence of an individual using our technology. So this is the cover of Nature, Your Life in Your Hands, where we describe the technology and describe the sequence of an individual using this technology. So in the UK, there's a big mission to exploit this technology. So we're following 100,000 people, we're sequencing them, we're following their health and the diseases they get in order to try and work out what the genetic contribution to some of the common diseases like heart disease, Alzheimer's disease, and cancer. So the number of human genomes, it's hard to see. This is 1999. Then we go to 2005, 2009, 2011, 2014. Hopefully you can see the number of human genomes sequences going up from 10 to 107 up to 18,000. And so the number of sequences is going up astronomically using this technology. So what I wanted to do in the last part of the talk is explain to you why this is important. So the first example is cancer. So here's someone unfortunate enough to have lung cancer. And so a sample, a biopsy is taken of the cancerous lung and the normal lung, and they're both sequenced and compared using the technology that I just described. And then once you compare the cancer with the normal uncancerous tissue, you can identify mutations. In this case, there's a mutation in a gene called RB1, which produces uncontrolled cell proliferation and cancerous growth. And often, once you know what's gone wrong, there's a treatment that you can apply to try and cure the cancer. Another example is rather than taking biopsies, there's a small amount of DNA that leaks out of the cancers. So you can just take a blood sample. There's a lot of DNA from normal cells, but if you sequence enough times, you can pick up the so-called circulating DNA from the cancer. And then you can identify again what gene has gone wrong, and design a treatment for the patient. This is another example. This is a little bit of a busy slide. So this is an example of a child who at 5 months had developmental problems and seizures. And in this example, they rapidly use whole genome sequencing. So the idea is you sequence the child and you sequence the mother and father, and you look for genetic differences. And so they found initially 13,367 differences, but some of these differences aren't in coding regions, so they have to be functional, they have to be linked to a disease, they have to cause a problem. And so you can triage, filter the differences, and they discovered that there's a variant in a gene called ATP7A that disrupts copper metabolism, and they could diagnose that the child had Menkes disease. And often, I think in about half the cases, once they've done this process, there's a treatment for the child, and the child can go home. And this has recently been is available in the NHS. So this is an example. This is from the BBC News site in 30th of September. This baby had a blood test because it was suspected to have a cancerous lump on its leg. And so they used a technique called whole genome sequencing, which you now know a lot about, to reveal that the mass was a myofibroma, and as a result, that the baby didn't need any chemotherapy and could come home. And so this is just one example of these tests being available on the NHS. This is another example where this child actually had a disease, he was sequenced, and they could come up with a treatment, and he went home with his parents. So I should say that this is well beyond what we imagined when we came up with the idea. We originally imagined people would use it as a research tool, not actually be used to actually save young children's lives. This is another example, maybe more relevant. So this is the number of sequences of COVID as a function of time. This is a logarithmic scale, and you can see this large increase in the number of sequences of COVID that are being produced. And this is important as the virus mutates to understand what the mutations are and monitor how it's changing. So again, this sequencing technology was used to monitor the changes in COVID. It's still being used. You can sequence sewage and look at the virus and look at how it's changing. And this is maybe an example you're less familiar with. This is these are Homo sapiens, and these are Neanderthals. And so the technology has been used to get little bits of DNA from remains of Neanderthals and these so-called Denisovans, and sequence them and understand the differences between us and Neanderthals. And this is an area called paleogenetics, and the Nobel Prize for Medicine was awarded this year for the person who developed this technology and exploited it to really understand how we evolved from Neanderthals to humans. And really, I'd just like to finish now. So I just wanted to stress a few points. Science is a social activity. The DNA sequencing wouldn't have been possible if I hadn't worked. I'm a physical chemist, I like building instruments. If I hadn't worked with Shankar, who's a chemical biologist and an expert in DNA and nucleotides. It's hard work. Like I tried to show you, often our experiments don't work. But what's motivating, I think, is we know we're doing something that no one's done before, and it's very exciting when you get it to work. And like I tried to show you, when you get it to work, it might have really important consequences. So you might be in the right place at the right time only once. And so another thing that's very important is to recognize when you come up with a good idea and act on it. So we didn't know if the DNA sequencing idea we came up with, which I've explained to you, would work or wouldn't work. There were lots of things that could potentially have gone wrong. But I think the thing we did well is at least give it a go and see if it was possible. And as I've shown you, that's been very exciting and gone off in directions that we wouldn't have expected. It's important to emphasize the idea came from basic curiosity-driven science. We weren't trying to do DNA sequencing, we're trying to watch the DNA polymerase. And this is an argument for the science funders to fund basic curiosity-driven science because it's important, it's a generator of these new ideas. And hopefully, lastly, I've shown you that new chemistry and new ways to measure things can solve major societal problems and improve people's lives. And I'll happily answer any questions that you have. Thank you.
Do we have any questions from anybody? Don't be shy. Oh, really? Over there? Come on.
What were the challenges when designing the initial radioactive labels to fit the template?
So, I mean, I think there were multiple challenges in getting the sequencing to work. In terms of the nucleotides, it was designing something that the polymerase would incorporate correctly, because there's no point in putting the color-coded nucleotides in unless it was the correct. If it was an A, it was a T. So that was so I think making them was it's not my area of expertise, but actually making the structures I showed you was the first problem, to actually make them and make pure versions of them in enough amounts that we could do the experiments. Then the next problem is ensuring that the pairing of the bases is correct. And to do that, we also had to mutate the polymerase so it could accommodate these altered bases. So there was a big program to do that. There was also a lot of work done in the surface chemistry, because our method relied on the method of the DNA, that color-coded DNA that we put in, the color-coded nucleotides that we put in being incorporated into the DNA, but if they stuck to the surface, we couldn't distinguish between stuck nucleotides and incorporated nucleotides. So we had to make these incredibly non-stick surfaces in order to get it to work. So those were the main challenges. And then once we had it working for one cycle, then we had to get it, it was straightforward to do multiple cycles, but we had to improve the efficiency so we could get out to 30 cycles. That was the minimum we needed to do to map back onto the human genome. So this series of problems, once we solved one problem, then we moved on to the next problem.
Magic. Any other questions? Cool. I'm going to be slightly lazy and go for the person in the middle.
So there were many examples of young children being diagnosed with diseases by DNA sequences. Is there any difference between DNA sequences in adults and young children?
There's no difference. I mean, the problem is that these children get born and they have a mutation that means that they can't breathe very well. And it's quite a big problem, because you need to treat them within a few days, otherwise they'll actually die. So that the person who developed exploited the technology in San Diego, I think holds the world record from getting the sample and sequencing. They did it in about 24 hours. And so once you have that information, you can very quickly come up with a treatment. But a few days matters in those examples.
Is that okay? How did you adapt your method to only sequence certain parts of the DNA if you just wanted to look for one gene, or just the coding areas?
So you can. Essentially, the idea of the method was we sequence the whole genome, but you can do so-called exon sequencing, where you just get the coding regions. So you can just pre-select the coding regions, and that's the sample you put in. But the whole idea is you sequence the whole genome, because there's a lot of discussion about how important the non-coding regions are as well. So the whole idea was to sequence the 3 billion bases. And the bits that we have problems are the repetitive regions, because it's very hard to reconstruct the repetitive regions. But but arguably, well, we would argue those are less important, but there is evidence now that they're important in terms of the overall structure of the DNA. But the driver was to sequence everything, because we don't know what's important and what isn't important. Certainly at the time, we now know a lot more, and you can sequence smaller parts of the genome by selecting before you put it into the sequencer. But still, I think that it's really important to sequence everything and get full information. So the vision would be that newborn babies are sequenced at birth. That costs $200, so that might not be so expensive. And that's something that would be invaluable in terms of designing treatments that are personalized for that person over their whole life. And I think we might be reaching a tipping point where it becomes economic to sequence newborn babies rather than wait till they're 40 or 50 and have some horrible disease and then try and treat them.
So we've got a question from someone online about, could you put other animal genomes into the human DNA? This is from Jacob Slido.
So you can certainly see that there's a big effort to sequence lots of other animals. I think there's a big effort to sequence all the animals in the UK, so we have a record of their genomes, particularly for species that are going to might be extinct. So there's certainly a lot of work sequencing things other than humans. I focus entirely on humans. In terms of mixing humans with animals, that's something that I don't think is ethically allowed. And certainly it's not something you can do with our technology. There's other technology, which is gene editing, that has the capabilities to do this.
Yeah, thank you very much for entertaining that question. Do we have any other questions? Ideally not human-animal hybrid based.
Hello. So what's next for your research and technology?
So we think we've handed over the DNA sequencing to the company who's done an excellent job at engineering it. And I think 90% of all the genome data has been generated on our platform. So I've largely focused on using the same techniques to understand what causes Alzheimer's and Parkinson's disease, imaging the aggregates, imaging the real aggregates that form in human beings to understand how they cause Alzheimer's and Parkinson's, and so we can develop new therapies, and we can also have early diagnosis. And like I alluded to, that looks quite promising, though we've got a lot of work to do. So we're basically using the same technology, but applying it to protein aggregates now.
Yeah, I was just wondering if there were any side effects or negative implications when you are developing this technology on humans.
So I think that the ethical issues that needed to be considered if you start sequencing people. So James Watson, when they developed the project to do the first average human genome, James Watson also considered the ethics about this. So what do you do if you have a newborn baby who's got a mutation that means they're going to have a disease that's going to require a very expensive treatment? What sort of can you make a decision based on their genome? And so I think that the consensus around this was you shouldn't do anything until they're over 18 in terms of using their genome information. But I think the answer is there's some serious ethical issues around what decisions you make based on people's sequence. I think treating people, like I've explained, I think everyone would agree are good things, but that there are ethical issues around making decisions based on people's sequence that are really questions that us as a society need to address, and particularly decisions about people getting treatment or not when they're very young, making decisions based on people's sequence. I think most people would think that we shouldn't do that. So there are some serious ethical issues if we were to sequence everyone. So there's a film, and maybe it's quite old now, called Gattaca, that came out maybe 15 years ago, that imagined a world where everyone had a sequence and sort of explored. People had good sequences, were sort of in much better positions than people who had bad sequences. And the film sort of discussed the fact that it's not all our sequence. Quite a lot of what makes us up is also environmental, and it's not all our sequence that makes our personality. But I mean, so that film, if it's still out, is quite a good exploration of some of the ethical issues around sequencing. Yes. So there's a chemical moiety that's there, and we add a reagent in water, and it removes that functionality and replaces it with an OH. Yes.
Would this sequencing be able to detect potentially terminal diseases and treat them before they cause death?
Yes. Yeah. I mean, I think yes. That's the idea. So the idea would be a lot of diseases take some time to develop, and you could sequence people, and if you had a treatment, then you could treat them. There's a problem around the disease. I work on Alzheimer's, that we could actually, and an ethical issue, that we could actually tell people that they were likely to get Alzheimer's disease, but there's no treatment. And so would you want to know if you were going to get Alzheimer's if there's no treatment? The idea would be we can work out what's going wrong, and that tells us pathways that we can develop therapies. But so I haven't been sequenced. My colleague Shankar's been sequenced. And one of these genes, it's called ApoE. He didn't want to know his ApoE status just because of the reasons I've explained. If you're ApoE 44, you're 13 times more likely to get Alzheimer's disease, and there's no treatment. So that's a difficult decision. I think you might want to know so you can put things in place as you develop the disease, but you might not want to know. And also, it's difficult because these are probabilities. If you have a certain sequence, it doesn't mean you're going to get the disease. It means you're more likely to get the disease. So, but I think we will be able to catch people before they get terminal diseases. But we need treatments as well. Yes.
Are there any risks of DNA sequencing?
Well, I think, like I've explained, the risk is making decisions based on someone's sequence. So some sort of discrimination based on people's sequence, I think, would be the risk. That if we were all sequenced, and then if you had a good DNA sequence, you got privileged treatment in society, I think that's the risk.